the work that I'm going to present will actually overlap a bit with what Trevor was talking about earlier. And I guess it came to my attention about a year ago, our attentions, that we'd sort of been doing kind of equivalent projects, which was a bit of a frightening sort of coincidence to realise. But on the plus side, it's good to be able to reproduce a result completely independently between different labs, which is a sort of, I guess, a gold standard for science research. So maybe I'll skip the first slide. And essentially then, so GABA receptors are heteromeric proteins, as you can see up in the top right corner. And typically they're 2 alphas, 2 betas, and a gamma or a delta subunit. And as yet, heteromeric GABA receptors have eluded structural determination, and in my experience at least, they're very hard to work with in terms of structural studies. But a few years ago, we managed to solve a simplified homomeric protein, which was beta-3 homomer, which was based on 5 beta-3 subunits. And that was missing the intracellular loop, but maintaining the transmembrane domain and the extracellular domain, which is sufficient for most of the sort of basic pharmacology of these receptors. And so obviously this gave us some insight into the structure of these receptors, but we wanted to learn more. And so one of the things we tried to look at was expressing alpha subunits as homomeric receptors. So this is this example over here is a gel filtration profile for the alpha 5 subunit. And gel filtration is sort of a quality control for deciding whether to proceed with crystallization trials of a protein. So you run the protein, you purify your protein, you run it down a size exclusion column, and you want to see basically a nice sharp single peak, as in this example here, which suggests that your protein is all in kind of one state and one size, for example, in this case, a pentamer. And for the alpha 5, obviously, we got this low yield and this fairly messy trace, which suggested it sort of maybe multimerized a little bit, but it wasn't forming a clear construct. So we decided to make a chimeric protein, which Trevor did, although Trevor's was with a bacterial extracellular domain from a bacterial protein, whereas in our case, we used our beta-3 construct. And so we have a beta-3 extracellular domain and the alpha-5 transmembrane domain. And this chimera gave us a nice monodispersed species, which suggested we could try crystallization. And when we expressed this construct in cells, we could get gating in response to the agonist. So this is histamine, which is the agonist for beta-3 homomeric receptor, rather than GABA, which is for the heteromers. But we had this large spontaneous current, which was blocked by the GABA blocker picrotoxin. So the receptor had a large amount of spontaneous gating. And this made it very difficult to work with. So we put a mutation into the receptor, and we targeted a loop that's known to be involved in gating. We managed to ablate most of the spontaneous activation, and then we could look at low-level submaximal activation by histamine, and we could see activation by the neurosteroid pregnenolone. And so the reason we were looking at this construct with relevance to neurosteroids was that, as previously mentioned by Trevor in one of his sort of landmark papers from about 10 years ago now, they showed that the alpha subunit was absolutely critical for neurosteroid binding. So we could get a dose-response curve for the potentiation by neurosteroid, and if we introduce one of the mutations that Trevor had identified, we could knock out the potentiation, suggesting that this site, not only do we have a neurosteroid site in our chimera, but this site is equivalent to the one that was in the heteromers. So one of the problems that we have when we were working with our beta-3 homomeric protein was that it was very difficult to crystallize. It would hardly ever crystallize. We got these fairly ugly crystals over here that aren't very sharp, strangely like weird butterfly things, and they would kind of diffract to low resolution. They would grow fast. And we were lucky that we got this one supercrystal, which kind of rose up like the Great Pyramid of Giza out of the solution. And you can actually see it's actually poking out the top of the liquid, and part of the crystal is actually dry, which is very bizarre. And that was what gave us our structure. So we wanted to address this and try and get much more crystals, much more rationally. So we collaborated with Jan Stout in Belgium, and we sent him some of our GABA receptor. He injected it into llamas and raised nanobodies, and we got 18 nanobodies sent back to us, which we assessed for binding. And so these nanobodies, essentially, they're like miniaturized antibodies. So we attached our GABA receptor to chips, and then we did surface SPR. And you can run the nanobody over the chip, and it will stick to the GABA receptor if it binds. And you can also wash it off. And so, yes. So we did some binding studies for various different nanobodies, and here we're washing on doses of nanobody, and we're getting progressively more and more binding, and the nanobody's basically not washing off or washing off pretty slowly, which is indicative of very high affinity. In this case, nanomolar. Generally, there are between 1 and 10 nanomolar. So these nanobodies really seem to like binding to the GABA receptors. And when we tried crystallization of the complex of the GABA receptor with the nanobody, we got many, many more crystals than we had before, more than we could pick and shoot, which was bad news for Radu, my boss, who was the one that was going in the cold room to fish all these crystals. But in the end, it gave us much better ability to determine structures. And so we can see a couple of examples here. Where we can see that so this is the GABA receptor. We have the transmembrane domains in red and the extracellular domains in blue, and the nanobodies in green. And so they're kind of wrapping themselves around the receptor, one binding per subunit, and they make contacts in the crystal lattice between the receptors. So they're mediating crystal contacts, basically, and therefore they're very useful if you've got a protein that behaves well but maybe doesn't crystallize well. It's definitely worth trying to generate nanobodies against your protein for structural studies. So we solved the structure then of our chimeric protein with this beta extracellular domain and alpha transmembrane domain. And we have our nanobody in this instance binding at the interface between subunits, just under this light blue loop here, which is key loop C involved in agonist binding, and this is the sort of neurotransmitter pocket. And as expected, the three variable loops in nanobodies, the complementarity determining regions, bind are the ones that are responsible for binding across the interface of the GABA receptor. So it's been suggested that nanobodies are very good at binding to sort of crevices and pockets, and this CDR3 loop kind of reaches in like a finger to sort of latch and attach to the pockets, and that's certainly what we've seen for these GABA receptors, which was maybe why with a protein that has these kinds of interfaces and pockets, they are good targets. So when we solved this structure, we could see that the extracellular domains, so in beige we have our original beta-3, and in blue we have our beta-3 from the chimera. We could see that the conformations were very similar. So that meant that the extracellular domain, which we knew from our previous construct, was in an activated conformation bound by agonist. So it assumed a similar conformation, and the transmembrane domains had assumed this characteristic, I like to sort of refer to it as a kind of a slouch, which is, I guess, a bit slangy. This kind of slouch where the gate is present down the bottom at this proline, and this is a desensitized conformation. As we can see here, this is the original beta-3, this is our alpha-5, and then here we've got what's known to be the closed glycine receptor, which is sort of straighter, would be straighter and narrower, and the open glycine receptor, which is straighter and wider. And so we also solved this structure in the presence of the neurosteroid. So maybe just I'll try and so we can see then the extracellular domains, we have our nanobodies binding around the outside, and in orange we have these sugars. And directly below the nanobodies, this is where the neurosteroid was bound. This is an endogenous neurosteroid pregnenolone, which is also bound, therefore, at the interface directly below the neurotransmitter sites. And when we observe the residues and the binding mode, then we can see that this key glutamine residue, which Trevor identified, is mediating the contact, a polar contact with this C3 hydroxyl at the top of the molecule. And there's this tryptophan which forms a stacking interaction against the rings of the neurosteroid, which is a key contributing factor, and tryptophans are one of the most commonly conserved residues in neurosteroid binding sites. And if we look at the ligand density, so if we fit the protein through the density, then we're left with this unaccounted for density here at the position for the neurosteroid site. And we can dock our pregnenolone very nicely in there. One of the nice things about pregnenolone actually is it has a natural kink in its structure, and this can allow this helps us to fairly unambiguously dock our ligand into the density. And so this was at about 3.2 angstroms, and across the 5 sites going around the receptor, the binding mode was basically identical between each of the sites. So if we consider this then in the context of the heteromeric receptor, so we'll have a number of different interfaces in the heteromer receptor. We won't have an alpha-alpha interface as we had in our structure, but we'll have a beta-alpha interface here, for example, and an alpha-beta interface here. And as we have a structure of the beta-3 subunit in the same conformation as our alpha subunit, we can simply swap in one beta subunit on one side or on the other. And what we can see is for the alpha-beta interface, the beta subunit introduces this tryptophan in place of the glutamine. So the glutamine is only conserved in alpha subunits. It's not present in the betas. And this blocks off the top of the site and will sterically hinder neurosteroid binding. Whereas if we look at the other interface, the binding mode will be preserved because the beta subunit maintains residues which permit and don't obscure the site in any way. And if we just do a mutagenesis, so if we then go back to the heteromeric receptor and we mutate this leucine, which is sort of sandwiching the neurosteroid against the tryptophan, then we see for 2 different neurosteroids that we tested, pregnenolone and allopregnenolone, that we reduce the sensitivity of the heteromer about 10-fold for neurosteroid when we introduce this leucine mutation. And another one we did, this phenylalanine, which is a little bit more peripheral, didn't have any effect. So we did a bit of computational modeling. And in yellow over here, we have our computationally docked pregnenolone versus the original structure. And we can see that the docking study that we did with Schrödinger gave close agreement. And then if we look at the 3 ligands again, so this is now pregnenolone again, we can see that the docking program placed this C3 hydroxyl sort of halfway between the glutamine and the tryptophan. And again, if we look at allopregnenolone, which is another endogenous neurosteroid, which is an isomer of pregnenolone, but doesn't have the kink, then the equidistance of this C3 hydroxyl is maintained between these 2 regions, and the rest of the receptor is sort of shifted down a little bit. And then if we contrast this to what's known as the 3-beta pregnenolone or epipregnenolone, this has the hydroxyl instead in the up orientation. And it's been shown a lot of times that if you change this C3 hydroxyl from a kind of a down to an up, then you significantly weaken and attenuate the potentiation process. And we can see that obviously this will sort of hinder this kind of equidistant positioning of the C3. So this raises the possibility, at least, that maybe this is the positioning of this C3 between these 2 residues is a significant contributing factor to whether the neurosteroid will potentiate or not. So we also then went on to solve the structure without pregnenolone bound, which is this orange structure here. So I've just aligned the 2 sites. In red, we've got the original one with pregnenolone bound and the pregnenolone shown. And then in orange, we've got the apo structure. And we can see that there's some subtle local impacts on the structure. And this glutamine is just very slightly drawn down towards the C3 hydroxyl whereas slightly lower down this M1 helix, the valine and the tryptophan are actually pushed a little bit away to accommodate the rings of the pregnenolone. And so these distances are quite small, they're about 0.7 angstroms. And if we look at the impact, it causes a kind of torsion on the bottom of this M1 helix, just sort of bending it away from the pregnenolone, which impacts on this very short 3 amino acid linker across to this proline, which lines the gate at the bottom of the pore. And so if we just compare the structures as a whole, then we can see that the two structures, the apo and the pregnenolone bound, are actually identical in the extracellular domain and also at the tops of the transmembrane domain. And the only difference is this small local change where the pregnenolone is either present or absent. And we can just see that here. It causes this very small effect on the diameter of the gate at the bottom, whereas the others are almost practically identical. And this is still a small effect. It's only a fraction of an angstrom. And compared to, say, a glycine receptor, which between its desensitized, its slouched conformation, and its straight open conformation, obviously there's a much bigger difference. But this sort of hints maybe that neurosteroids sort of pull at the desensitization gate to try and withdraw it and maybe just increase the probability of the receptor reverting back to the open conformation. So maybe I can just, again, try. So we have the neurosteroid binding, we have the glutamine moving towards the neurosteroid, and this helix, the M1 helix, twisting away. And then this pulls on the pore to cause a slight widening and a slight, I guess, torsional effect on the M2 and the bottom of the M1 helices, which we can just see again here. And just one last time from the side, so we can see that the helix undergoes a slight stretching and bending outwards at the desensitization gate. So at the moment, this is still a hypothesis. Nevertheless, there has been some data that was produced a while ago which actually showed that if you recorded from GABA receptors in patches and you applied a maximal dose of GABA, and then you looked afterwards at the ability of the GABA receptor to spontaneously flicker and reopen after the exposure to the GABA, that in the presence of neurosteroid, you got a lot more of this reopening activity. So that kind of fits with the structural data, at least, that we have, which suggests maybe the neurosteroids can pull on this desensitization gate and encourage the receptor to reopen in response to having desensitized, and therefore generally potentiate the, and maybe that's their mechanism of potentiating activation and opening of the receptor. So I just need to acknowledge my overlord, Professor Radu Arăcescu, who periodically provides me with inspiration to keep me moving forward. And then our collaborators in Belgium who produced the nanobodies, the Luigi who did the docking work, Simonas who made the nice movies that I showed you and also did a lot of work on the nanobodies, and Suzanne Scott who did generally was involved in all aspects of the wet lab work. And that's it. Thank you.
Hi, Paul. Great story. So you're sort of alluding to the end with Stefano Vicini's work about how maybe it works, but could it be that what it's doing is actually stabilizing the agonist, and so it's prolonging its ability to actually act as an agonist? So it's do you see what I mean? So there's two ways to think about it. It's doing something itself, or it's actually helping the agonist do its job there.
Yes. Well, I guess if
maybe that's the same thing, but structurally it would be different.
So I mean, I think obviously GABA receptors in the open state will have a higher affinity for the agonist. So if the pregnenolone can interact with the receptor to support the open state, it will increase the affinity of the agonist and therefore prolong the binding event as well. So I guess it will, this is kind of obviously a global effect. What we're seeing here is a little localized effect, but it will be obviously more complicated, in the real thing when everything's happening together, definitely.
do you think the small movements that you see might also contribute towards direct activation of the receptor by neurosteroids? Or do you think that's reserved solely for potentiation?
So I think what we're seeing here is a small effect, and I think if you're able to look at it in the context of a bilayer, because this site is very shallow actually, so the neurosteroid will make substantial contacts with the receptor and with the membrane. And I think if you could look at it in a bilayer, you'd see a bigger pull on the base of the transmembrane domain. That's my kind of hypothesis and my feeling. So I think obviously it would be one of the things that can drive the direct activation. I guess activation is maybe when all the sites are occupied or enough sites get occupied. But when you only have one, then it's instead it's more of a potentiating effect, and it requires GABA as an energetic factor to drive the opening.